Hello, dear friends. In last lecture, we have discussed about the experiment of Griffith, which proved the DNA is a genetic material that can be transferred from one cell to another. And we have uh, explained the experiment carried by Griffith for proof in that experiment. Today, in this lecture, we will discuss about the another scientist, that is Avery, Macloed, and MacKarthy. These are the three uh, scientists who perform another experiment for supporting the experiment performed by the Griffith, and they found the somewhat uh, similar type of result, and these results are helpful for the Griffith experiment to confirm that DNA is the material that is responsible for the transformation of non-virulent strain to virulent strain, and that transformation or transfer of genetic information from one cell to another. In the Griffith experiment, we have seen he used two strains of pneumonia. One is a virulent strain, and another is non-virulent strain. So, Diplococcus pneumoniae AIV or Streptococcus pneumoniae. So, S type of strain, R type of strain. So by using the similar type of strain, that is R strain and RES strain. In last lecture, we have already discussed about the what is meant by the R type of strain and what is meant by the ES type of strain. R type of strain is a non-pathogenic or non-virulent type of bacteria, and ES type of bacteria is a pathogenic or virulent bacteria. So in this lecture, we will discuss about the experiment performed by the Avery, Macleod, and McCarthy. So in the screen, we can see here are the different uh, images that are the petri plate, which shows the different types of color type are red color type of colony. These are the bacterial growth occur on the uh, petri plate. So we start the lecture here. After the completion of Griffith experiment, it is not concrete evidence that the DNA is a genetic material. It is transfer genetic information. So, for the complete concrete evidence, every Macleod and McCarthy they proved a solid evidence to prove that DNA is the material. It acts as a genetic material. So, for that, he used two types of strain. It is R type of strain, that is non-virulent strain, pneumococcus strain, and another is ES type of strain, that is virulent strain. So in this first experiment, he performed, they use type R type of cells that are non-virulent, and they inoculated that non-virulent type of cells on the nutrient media, and he found that the R type of cells easily grow on the nutrient containing media and shows a normal colonies of R type of bacteria. Later on, they again inoculated only the DNA from the hit kill type of ES type of cells. The ES type of cells which are treated with the or they are given with a heat treatment so that cells were live and only they represent nuclear material, and they inoculated the DNA from the hit kill ES type of virulent strain on the nutrient media. But there is no viable colony, so that due to the heat treatment, the viable cells are killed. The only genetic material is present there, and that genetic material is not able to grow on the nutrient media. So that plate shows no type of or shows any kind of didn't show any type of growth on the nutrient media. This is a second step of the experiment. Here the primary stage where R type of cells were inoculated on the plate. It there shows R type of colonies, In second step, um, uh, they use hit kill yes type of cell, and they show no growth on the nutrient media. Okay. Later on, they take a type R type of cells, that is non-virulent R type of cells. These are plus they also inoculated the DNA from the hit-killed ES type of cells and mixed them. Means R type of cells plus DNA from the ES type of hit-killed cells plus serum that precipitate R type of cells from mixture. So they precipitated the mixture and they took a serum that precipitated R type of cells and again inoculated that mixture on the nutrient media, and they found there is again a yes type of colony. Yes type of colonies are virulent strains. That means when you take only hit kill DNA in second stage you didn't observe the yes type of colony, there is no growth. But when you take R type of cells plus hit kills yes type of cell and with the R type of mixture, you find yes type of colony. Okay. Later on, again, they repeat the same type of experiment with R type of cells plus DNA from the hit killed yes type of cells. That means these two stage or these components are similar plus serum that precipitate uh, R type of cells from the mixture. Again, the stage is common plus they mix that mixture with the protease. Protease is an enzyme which degrade the proteins. So it is nothing but a protein degrading enzyme and inoculated that mixture on the nutrient agar plate. Again, they observe yes type of colony. Here they use protease because they are not sure about the whether the protein is transferring the S type of characteristics to the R type of cells, so that S type of colonies are observed in the third experiment. So for that they use the protease. They assume that there may be a protein particle that transfer the S type of colonies or S type of DNA to the R type of cells and observing S type of colonies. So for that, to prove that experiment, they use protease, but they failure in this experiment. That means protein are not a molecule that is transferring the S type of characteristics to R type to cell or R type to S types, and they are observing the S type of colony. So again, they use similar process, that is R type of cells plus hit killed DNA from S type of cells plus serum that precipitate R type of cells from mixture plus RNA. So they assume that if there is no if that particle is not protein, maybe RNA particle. So they use RNA. RNA is again is enzyme which break down the RNA molecule and inoculated that mixture on the nutrient agar plate. So again after that they found again yes type of colonies on the media. That means again their assumption is disproved. That is RNA is not that particle which is uh, transferring R type of cells into the S type of cells. So after that, in next step, they use R type of cells plus 8 kill DNA from the S type of cells plus serum that precipitate R type of mixture plus DNA. So DNA is again another enzyme which break down the DNA. So they use DNase enzyme. After the treatment of DNA, again they inoculated that mixture on the nutrient agar plate containing the same type of media, and they observe there is no growth of any kind of S type of R type of colony. So according to their assumption, either maybe protein or RNA or DNA are transforming the R type of cells into the ES type of cells of pneumonia, um, is responsible either protein, RNA, or DNA, and they found that the transforming principle is nor protein or either a RNA. It is DNA. But that experiment is not concrete evidence. So for complete concrete evidence, they again perform a similar type of experiment. They use R type of cells plus purified ES cell polysaccharide. Polysaccharide is nothing but a capsule content of the ES type of cells. Polysaccharide are present in the or already pneumococci is a capsulated bacteria. So they maybe assume that maybe the capsule is a molecule that is transforming the R type of cell into S type of cells. So they use capsule component of the capsule for the uh, conversion of R type cell to S type of cells, and they found a negative result. That is, there is no transformation of R types of cells into S type of cells, and they uh, observed only R type of colony. So polysaccharide is not a transforming principle. So again, they use R type of cells plus purified ES cell protein. So again, in second step, they use R type of cells that is non-virulent plus purified ES cell protein. Maybe protein molecule is a transforming principle. So they use S type of cell, S type of cell protein, and they again found negative result, and they observe R type of colony. So result are similar for polysaccharide and protein. So protein is again not a transforming principle. In third stage, they use R type of cells plus purified ES cell RNA. So that mixture is inoculated again on the nutrient agar plate, and they observe R type of colony. And again they got a negative result. That means RNA is not a transforming principle. It's converting R type of cell to the S type of cell because they inoculated R type of cells, and they observing the R type of colony. Purified uh, live cells of R cells plus purified extract of S type of cells proteins are inoculated in third stage. Okay, there is no presence of live S type of cells, only the component of capsule, protein molecule, RNA from the S cells, DNA from the R cells are inoculated or mixed with the live R type of cells, and they observe the growth of R type of colonies in all these three stages. In fourth stage, they use R type of live cells plus purified S cell DNA, that is non-virulent R type of cells, live cells plus purified DNA from the S type of virulent strain, and inoculated on the media. They observe S type of colony. Means R type of cells are transformed to S type of virulent strain. That means our non-virulent strain is transformed to S type of cells. And this is again a concrete evidence proving that the DNA molecule is the molecule converting the non-pathogenic or non-virulent strain to the virulent strain. Again, yes cell extract, they, for confirmation of that experiment, they use S cell extract. That means S type of cells extract plus protease plus R cells, that are live cells. Again, they treat the S type of extract with protease. The protease is again a protein degrading enzyme and inoculated, and they observe the S type of colonies. Means protein is not a molecule that is transforming R type of cell to S type of cells. Again, they use S type cell extract plus RNAs plus R cells. That means only the cell extract of S type of cells, that is virulent strain, plus RNAs, that is RNA degrading enzyme, plus live cells of R type of non-virulent strain and inoculated on the plate, and they observe S type of colonies. So again, these evidence are supplementary with the experiment carried out by the Griffith and experiment of primary experiment of Avery, Macloed and MacKarthy, and prove that the DNA is the molecule that is transforming S type of R type of non-virulent uh, pneumococcal strain to the S type of virulent strain. That means DNA is the only molecule that is transforming a non-virulent strain to the virulent strain, and is hence they prove the experiment performed by the Griffith is true in all aspects. So this is all about the uh, experiment carried out by the Avery, Macloed and MacKarthy and Griffith to prove that DNA is the genetic material that is responsible for the transformation of one non-virulent strain to the virulent strain, or carry the information from one cell to the another cell. So this is all about the